Hello and welcome everyone. I am Rifal Jagil. I'm your moderator for today's presentation. I am the Field Application Sciences Director at Unchained Labs, and I am exceptionally excited for this part 3 of this virtual series that we've been doing. So in part 1, we told you about the fluorescence detector of the uncle and exactly what makes it unique, the ability to do intrinsic and extrinsic fluorescence. In part 2, we covered a very special topic regarding blue shift, right? And part 3 is actually quite nice because we round everything up and we're going to be discussing essentially the benefit of having all 3 detectors and the benefit of being able to use all 3 of them at the same time. And so for this presentation today, we have Kevin, who is our marketing director for the analytical product, and he'll be walking you through the uncle. So I am your moderator today, so I'm going to ask you to look down at the bottom of your screen or at the very top, depending on your panel, and you'll find a Q&A icon. So if you have any questions, feel free to enter them there directly. There's also a chat icon. So if you have any questions, feel free to post them directly there. We'll answer them. But certainly if you have questions and you want us to answer them, I recommend doing the Q&A and we'll answer as many of them live as we can. Otherwise, we'll follow up with you afterwards. So I'm going to hand this off to Kevin and enjoy. Talk to you soon.
All right, thanks, Rafar. I too am exceptionally excited. So here we go. Let's talk about combining tools for new insights on uncle. All right, so sometimes if you only look at things one way, it can be unclear to understand exactly what you're looking at. So that's why it can be helpful to take a different approach, different angle, get more data to see what you're looking at and really understand what you have in front of you. So that's when it's time to meet our uncle. Which is the all-in-one biologic stability platform because it combines 3 different detection methods, which are full spectrum fluorescence, static light scattering, and dynamic light scattering, that gives you a lot of different looks at what's going on in your sample so you can really understand the behavior, the stability, the aggregation, everything that's happening in there related to conformational and colloidal stability of your proteins or antibodies, viral capsids, things like that. So uncle combines those 3 detection methods across 12 different applications and then only consumes 9 microliters of sample anytime it wants to run any of those 3 detection methods on each sample. Importantly, it also includes temperature control and lets you look at labeled fluid measurements. So we'll spend a little bit more time looking at each of those during this presentation. So uncle's 3 detection modes that I mentioned, going a little bit into more detail, are the full fluorescence, which excites with 1 or 2 lasers, either 266 nanometer laser, which is the UV laser, or the 473 nanometer laser, which is the blue laser. Once we excite with those 2 lasers, then uncle reads full spectrum fluorescence to give you the whole picture of the fluorescence going on in your sample, and that can be done with or without labels, so you can answer many different questions depending on what your experiment calls for. SLS is static light scattering, and that's going to be helpful to see aggregate formation at the same time as gathering fluorescence data. So uncle is the only system with fluorescence and SLS, and it does come standard just right out of the box, and it applies to the highest and lowest concentrations that uncle can read. And we'll take a look at data from both the highest and lowest concentrations that uncle can read today. DLS is dynamic light scattering, and it's the classic technique to crank up the detail on size and size distribution. So you'll be able to look directly at what the hydrodynamic diameter is of your sample as it's floating around in its buffer. And lastly, all of these techniques can be applied across temperature control, whether that be room temperature, elevated isothermal temperature, linear thermal ramps to stress out samples, or any of a variety of other, we'll say more exotic thermal ramp profiles to really see things like thermal recovery or isothermal experiments, where we do stepped ramps over time, to really understand what's going on in your sample. Okay, so the unique sort of secret sauce of the uncle is the consumable, the uni, where we have these tiny quartz cuvette wells that help tell this big story from the uncle. So a uni is composed of 16 9 microliter quartz cuvettes that are held in this aluminum frame and then sealed in this blue frame with silicone gaskets on either side. So because the samples are always sealed, there's no extra steps to adding extra sealant, and it's always really easy to pop in and out of an incubator or freezer to do freeze thaw studies or to do long-term elevated temperature isothermal experiments. So when you read your sample in a uni, what you'll get is typically a picture that looks something like this, where here we're taking a look at the thermal stability of a protein, and we're gathering information on both fluorescence, shown in blue, and SLS data, shown in green. So we'll go a little bit more into the fluorescence and SLS data and how this analysis works out, but suffice to say that in this analysis, we're looking at sort of a center of mass readout here on the y-axis for fluorescence. And as the signal goes through inflection points, indicated here as TM1 and TM2, those are melting events in your protein. The SLS data shown in green, it's more to do almost with the turbidity of the sample, or I always compare it to shining a flashlight on a foggy day. The more fog there is, the more signal you can see bouncing back at you, the brighter the flashlight beam is shown. That's much like how SLS is behaving here. So we have not much signal being bounced out until aggregation starts to occur, things have become cloudier, and lots and lots of SLS signal is present. Now, kind of where UNCLE gets really combines these two techniques and demonstrates a lot of value is the ability to see stability and aggregation in one experiment. So in this particular protein, we know that aggregation behavior is linked with TM2 rather than TM1 based on the temperatures that both of these onset at. So how UNCLE makes use of protein fluorescence is by exciting with the 266 nanometer laser, and then that laser will hit these three aromatic amino acids inside of proteins, tryptophan, tyrosine, and phenylalanine, and that will be absorbed, which is shown in the blue reference spectrum here, and then those amino acids will then emit fluorescence, which is shown in green of each of these amino acids. So combine all together, those amino acid fluorescence behaviors all add up and become the intrinsic protein fluorescence that you see in your protein. As proteins unfold, then the fluorescence behavior of those amino acids changes, whether it's because they're more solvent exposed now, or whether because their perhaps quenching environment has changed. It's really something you can answer once you've seen the full spectrum fluorescence. So here's a look at kind of the complete story of fluorescence as a protein unfolds and is heated up. So in this case, each one of these lines is a full spectrum fluorescence read from about 15 to 95 degrees Celsius. So as we start, we can see first, very obviously, the signal decreases. And then around maybe perhaps here, that's when we see, by eye, we can watch the peaks shift to the right. So when we do the analysis for this, this signal, what we'll be looking at is tracking the BCM, which is the weighted center of spectral mass wavelength. And that wavelength will also show a shift to the right at that same point. And it's a very sensitive technique since we're looking at the full area under that curve when we're doing that shift sort of analysis. So this is how UNCLE gathers raw data and then outputs that fluorescence data that we've seen on the previous slide. So importantly, because we're gathering full spectrum fluorescence, we also get to focus on just the signals that we want, so we can focus in on protein fluorescence, maybe when we have a fluorescent buffer component present, or we could add dye and focus and answer kind of a different question. So with Cypher Orange, it's again looking at protein unfolding through a different way. And that might be one way that you help to do a bridging study between a Cypher Orange experiment you're doing in a qPCR plate and an intrinsic fluorescence experiment where you're not going to have additional dyes that interfere with your protein behavior. Likewise, ANS is another method of boosting your protein signal and adding fluorescence to it as well. And then CPM is another interesting dye that can be used to look at membrane protein stability since it looks specifically at cysteine bonds. Likewise, Cybergold is also another dye that we've been using a lot lately to look at DNA release from viral capsids. So only UNCLE really lets you look at this full spectrum fluorescence and gives you this kind of flexibility with how you design your experiment and additional flexibility in what kind of questions you can solve. Again, with full spectrum fluorescence also comes analysis. So our analysis package that we offer contains 4 different analyses right out of the box. The one that we prefer to use most of the time is this barycentric mean analysis because, again, it uses the whole full spectrum analysis and then divides that wavelength equally in half. And that gives us a very robust signal to noise ratio since we're using most of the spectrum. Likewise, we can also look at pure area under the curve analysis or a more traditional 350 over 330 ratio analysis or peak height analysis as well. So that's how we get to our unfolding data from our raw full spectrum fluorescence. Okay, so now we're talking about combining tools on the UNCLE. So let's shift over to static light scattering and talk a little bit about how UNCLE gathers that data. In this case, what UNCLE is doing is shining its 266 nanometer laser, could also shine its 473 nanometer laser, at a sample, and it's gathered by its fluorescence spectrometer. The intensity of light that this spectrometer is going to see is proportional to some optical constants, and then most importantly, concentration of the sample and molecular weight of the sample. This is the refractive index dependence on concentration, which for most proteins is constant. And I also want to comment that this equation has a lot of constants that are assumed, like light, laser intensity, distance to the laser, and angle, which on the UNCLE don't change. And there's some other assumptions that are held constant as well. But for this part, this gives us a nice jumping off point to look at both concentration and molecular weight and how SLS can help you look at those different parameters in your sample. So I mentioned that SLS on UNCLE is a out-of-the-box capability that applies across its full concentration range from low, which in this case, we're looking at 50 micrograms per milliliter of an antibody, and getting both an intrinsic fluorescence melting temperature and a very strong aggregation signal from SLS. So that's on the low end of the spectrum that we can still see aggregation data. And then on the high end of the spectrum, we can also still see intrinsic fluorescence, where we're looking at, again, an antibody from the same antibody, in fact, from concentrations of 150 mg per mil all the way up to 350 mg per mil. And then we notice that SLS, again, stays very consistent and helps you look at that aggregation behavior at those very high concentrations. So you might notice that that shift is present in the intrinsic fluorescence. We'll take a look at that in a little bit more detail in just about 2 slides. But first, because it's interesting, I want to show you what happens at high concentration. Well, because we have so much protein present in the sample, we have a lot of things going on. But what we can do is analyze this fluorescence signal for BCM, and then we know that the fluorescence signal kind of, we'll just say goes crazy for now. We do have a future talk coming up on high concentration protein samples and how you can gather data from them and what they mean. So in this case, we have fluorescence data that we can analyze, but what I want to highlight here is the SLS signal at 473, which you can see increasing just before that fluorescence signal sort of increases as well. So that's how we're looking at our SLS signal at 473 to gather really robust data on a high concentration protein sample. And in fact, if we compare our low-end concentration sample versus our high-end samples on the previous slides, we can see a shift in behavior of this antibody. So in this case, at low concentrations, we're seeing a melting temperature at about 68 degrees Celsius, followed by an aggregation temperature a little bit later on. However, once we increase concentration up to sort of the extremes of the solubility limits, we start to see that the behavior flips. So in this case, now we're seeing that Tagg is at much lower temperature, around, we'll say 60 degrees Celsius, while Tm is close to the previous temperature and then may be increasing as we increase concentrations. So this is actually really interesting because if you're not gathering both pieces of data at once, or maybe you're not able to gather both pieces at once because of limitations of instrumentation, then you're going to miss this flip in signal, and you may continue looking at melting temperature when in fact aggregation onset has already occurred at high concentration samples. So we mentioned concentration in the last few slides. Molecular weight is another parameter that static light scattering can help you look at. So in this case, we're looking at it in terms of sort of resolution of different samples. In this experiment, what we're doing is we're mixing different concentrations of lysozyme, hen egg white lysozyme is HUE, and bovine IgG to simulate changes in average molecular weight of that total sample. So what we can see on the left is as we increase concentration of the much, much larger IgG, then the signal intensity and the area under the curve is how we analyze this signal, increases as we increase concentration. Now we know that when we're increasing concentration, we're also increasing the average molecular weight of the sample, so we can compare the SLS signal versus the change in average molecular weight or mean molecular weight and see statistically significant changes in those different samples. So this is a really nice look at just how SLS can also be used to detect changes in aggregation and kind of what that really means on the biophysical level in terms of changes on the order of 15 kilodaltons or less. Okay, so that's a pretty cool look at basics of fluorescence and the basics of static light scattering. Well, let's take a little quick look at how real antibodies and real data look. So in this case, we have a sample of adalimumab where we see in green fluorescence and in blue SLS. So here we see two melting temperatures, but we also know from the SLS signal that the aggregation of this protein starts at TM1. So that's great because it indicates that we probably want to pay a lot of attention to TM1, and we'll probably view TM2 with a bit more skepticism since it's probably related to a biophysical event, but it's not a pure melting event. Likewise, if we look at a sample of trastuzumab in its commercial formulation buffer, we notice that this behaves a little bit differently. And in fact, this is the example from earlier where melting temperature 1 does not cause an aggregation event, but melting temperature 2 does lead to a Tagg. So melting temperature 2 might be something that we pay a lot more attention to during our screening analysis for formulations or mutants of this antibody. And in the third case here, we're looking at a cetuximab sample where we have Tagg occurring before TM1 or TM2. So then we know that Tagg is really the metric to pay attention to there, because that's how we'll be screening our samples relative to each other. So taking a look at all of these samples, we can then just sort of take a step back and understand this is how we use UNCLE to screen for different formulations conditions in this instance. So we see many different melting behaviors or aggregation behaviors, and then we can apply the same sort of approach to screening for many different antibody constructs. Okay, so we've talked a lot about monitoring two things so far in this talk, TM and Tagg from fluorescence and SLS data. So that's a good start to tracking antibody and formulation behavior. Two things is a pretty good beginning. But really, we want to have our eye on everything and keep an eye out for even more to solve more protein stability problem. So that leads us to dynamic light scattering, which is UNCLE's third detection method. So in this case, we have the ability to both do single sizing methods, say as a quick DLS read or maybe before a thermal ramp, or we can apply DLS across the thermal ramp and again use as another way to compare formulations or antibody constructs. As a quick primer for what DLS looks like on the UNCLE. So here we're having in blue, we have a nice monodisperse, happy, as I like to say, protein sample where we have a Z-average diameter and a PDI, where the Z-average diameter is what we expect for an antibody, and a PDI is a nice small value indicating a sharp peak. As the sample denatures, what we're expecting to see is a small increase in Z-average diameter and then an increase in PDI as well. And then as we continue to denature the sample, we'll actually start to see aggregate peaks appear as the average diameter shift even further to the right, as it's a weighted average diameter, and PDI increases even more. So that's the kind of information that you can get very quickly with dynamic light scattering, as well as information about each of those individual peaks shown in these graphs as well. So how do we use that during our melting temperature, our thermal stability, our colloidal stability experiments? So the first such example is to confirm what's happened after a ramp. So this is a pretty typical example where we've taken our 0.05 mg per mil antibody data, and we see aggregation by SLS. And if we look at DLS, we can compare the before ramp DLS read to confirm, yes, that sample is very nicely monodispersed prior to our ramp. So we should trust this data. And we can take a look at the sample at 95 degrees Celsius and see that, yes, aggregation has occurred. And in fact, we can quantify it and put a number on what that average diameter is there. So where this comes in really handy is checking quality before or maybe after a run. So I mentioned before already where it's knowing that my sample is not aggregated and ready to go. But let's say I had a group here that contacted me and they were using lysozyme as a control for melting temperature. So this was fine because, as you can see, their lysozyme samples were very reproducible, lining up right on top of each other, and they were very happy with the melting temperature behavior. However, they didn't see any Tagg behavior, and they were very curious about what's going on with their SLS, if it was working properly, is my concentration too low, does my sample not aggregate? And if you have no more data, there's nowhere else to turn to get the answer to these questions. But thankfully, dynamic light scattering is a pretty seamless inclusion into the workflow of TM, Tagg, DLS experiments on UNCLE. So in this case, what we did was we flipped over to the DLS tab to take a look at the data, and there was their problem. So lysozyme should be expected to show up around 3 or 4 nanometers with a very nice sharp peak. However, in their case, it was lots of aggregates. So their lysozyme that they were putting into their instrument was not where they expected it to be, but in fact at hundreds of nanometers in diameter, which tells them that that static light scattering information is not going to be valuable because it's already aggregated when you started. Okay, so that's a picture of how DLS can get you a true size answer and how DLS can help you troubleshoot an experiment that maybe didn't go quite the way that you expected it. And then here's a quick, another quick quantitative look at how DLS can work out during a thermal ramp, where we start with a monodispersed sample, and then at each one of these points, we're taking another dynamic light scattering read. We can look at the data from each of these points independently and watch how that PDI increases, the Z-average diameter increases, and that's going to indicate when our sample aggregates and when stability is a problem. If we take a look at this through a quick example of a formulation study, we can see that by adding increasing concentrations of arginine, so going from green to blue to gray, we've actually inhibited aggregation in this sample, which is kind of what we expect from adding high concentrations of arginine. All right, so in addition to DLS, SLS techniques with the DLS optical setups also let you peek at some interesting protein-protein interactions, which is great because it's something you can only get from having a true SLS optical setup. So this lets you go further in your formulation characterization to look at protein-protein interactions and understand if that formulation and protein combination is at risk for aggregation. So in this case, B22 and KD are designed specifically to look at increasing concentrations of your protein. And as you crowd them together, how does their behavior change? So let's start with the red line here. If we're looking at this sample, and as we crowd them together, we can think about the diffusion coefficient here, D first. As we crowd them together, if they act slower and slower and slower, then we conclude that their interactions are attractive, which is not desirable because it usually means it's going to lead to aggregation in your sample. On the other hand, if as we increase the concentration and increase the frequency of protein interactions, the diffusion coefficient increases, then we know that we have repulsive interactions. That's probably a good sign for our aggregation behavior. It's going to be harder to happen when you have repulsive interactions. B22 is a similar sort of philosophy, but we're looking at different metric on the y-axis. It's more analogous to the inverse of molecular weight. So in this case, if our molecular weight is increasing, our slope will be negative, and if our molecular weight sort of understanding decreases, our slope will be positive. So this is saying my sample looks smaller as I increase concentration, or my sample looks larger as I increase concentration. Again, very similar in terms of understanding how my proteins are interacting with each other. If we look at actual data, this is B22 data, we can see that for this protein in its native formulation, we had excellent positive B22 values, and this indicates repulsive interactions which are desirable. Adding sucrose to the formulation improves it very slightly, but adding arginine or sodium chloride basically ruin everything. So in this case, our B22 values have decreased significantly, and then we're now more close to a neutral slope, so we can expect that this sample will behave basically worse in an aggregation experiment. So that's how, with true SLS optical setups, you can get additional data from B22 as well as KD from dynamic light scattering. So we invite you to see things from all angles, and that includes your protein stability problems. And with that, I think we have a few questions coming in the queue. So I'll pause. Thank you for your attention, and let's handle some questions.
Thank you very much, Kevin. So that was great. So justifiably, I was excited. So let's get to this. So I'm just going to recap really quickly to make sure that we're on the same page, right? So UNCLE, full spectrum fluorescence. You told me that I have 3 detection methods. So fluorescence, SLS, and DLS. Sample holder is a special sauce, right? So small volume, 9 microliters. So that's fantastic. And then you also kind of went into your results by showing me that I can do high concentrations. I can also differentiate samples by Tagg when TM isn't sufficient, but I also have the ability to use DLS not only for sizing, I can do thermal ramp with it, but I can also do B22KD using your SLS detector. That sounds about right?
Yeah, it's a lot of options to solve a lot of problems.
Okay, that's great. I think that's really where I was excited about part 3. I thought this was a very nice way to bring everything together, so I think that's fantastic. It looks like the group is also excited. We have lots of questions. So I'm going to start with number 1, which says, if my protein cannot be differentiated by TM, are you aware of any example where TI could be used?
Yes, definitely. So it's kind of one of the classic examples we love showing on our slides. So this is a case where 2 proteins have the same melting temperature, but if you switch over and look at aggregation temperature, then we have dramatically different aggregation temperatures. So it kind of goes back to those earlier examples of maybe your protein aggregates at TM2 instead of TM1. So that's a very quick example of when TAG can add data to TM.
Excellent. Question number 2, are there any times that you can use all 3 detection methods at the same time?
Yeah, so we have a application in our isothermal application that allows us to basically alternate between reading fluorescence and SLS data and then going over and reading dynamic light scattering data on the same sample. So that enables you to understand both conformational stability and colloidal stability over long periods of time, up to a week in a uni. That helps you understand maybe how you want to design a months-long experiment, because you don't want to start that months-long experiment and see, oh no, I started too hot, all my samples are aggregated. So it's a really nice way to get data from all 3 at once.
Makes sense, excellent. Let's see, question number 3, can you do long-term isothermal studies?
Yeah, so as I mentioned, up to about a week, and then you sort of get to pick the resolution that you sample, that you read your samples there as well. So that's really nice because the uni's sealed, so you can do it up to a week in the uncle, or you can take the uni out after reading it once, put it in an incubator, and store it for longer, and then bring it back in. Same idea for freeze thaw studies too.
That's actually quite nice. Okay, awesome. Let's see, since you can do intrinsic fluorescence and extrinsic fluorescence, could you do them at once, at the same time? I'm assuming that's what they mean by that.
So yeah, so on the same sample, yeah. So the option to do that is, there's kind of 2 ways to do it. It's answering, well, we have 2 lasers. Do we turn on one or both? And it's very possible, say, for example, with Cypro Orange, to turn on both lasers and look at intrinsic fluorescence and Cypro Orange side by side. And the only word of caution that I have for that is just make sure to check if your dye is excited by the UV laser, because then you probably only want it to be excited by 1 laser at a time. So you only turn on the UV laser.
Very good. Let's see, question number 5, I believe now. So can you do SLS to determine molecular weight?
So what I would probably recommend is the best way to determine molecular weight is probably a B22 experiment where we're looking at SLS over multiple concentrations. It's a little bit like a Zim plot, but we're only looking at a fixed angle. So that's the same idea. The y-intercept helps you understand molecular weight. On a more practical level, dynamic light scattering is a really fast way to look at size in solution. So that's going to give you, probably the same kind of information that you're thinking of. Is my sample aggregated? How big is it? It gets you that answer in about 30 seconds.
That works. Excellent. Okay, so let's see. Next question. You say that your laser for intrinsic fluorescence is 266 nanometer. Isn't tryptophan excitation usually done at 280 nanometer? How do you answer that?
Yeah, so classically 280 nanometers did make sense for tryptophan excitation if you were only looking at tryptophan excitation, because that's where the absorbance peaks. So if you're looking at all of the amino acids of your protein, then 266 makes a lot of sense, because then you're able to capture all of the tyrosine emission as well. If you excite at 280, you're going to be exciting right in the middle of that tyrosine emission spectra, and you're not going to capture as much signal. In addition to that, 266 also gives you a really nice, clean SLS signal, since it's away from any of those emission spectra.
Makes sense. So basically, proteins are made up of more than 1 amino acid, and therefore you should look at all of them.
Yes, more data.
That makes sense. Awesome. Okay, let's see. So the antibody formulation screening experiments were done at 0.5 mg per ml. Does the experimentation and analysis change significantly at high concentrations, so 50 to 100 mg per ml?
So at high concentrations, I would say you still you run the same experimental design, and then you probably take a very careful look at the analysis, and you want to make sure that your aggregation behavior, where that occurs, you want to check that versus your melting temperature. And then you also want to check again for aggregates. So that's always kind of the classic example here. It's why you run DSC at very low concentrations. It's because you don't want aggregation to form. So that's again where that DLS example comes in. So I would say the experiment setup is the same. The analysis is very similar. And then just take all your due diligence steps when you're analyzing your sample.
Awesome. I'm going to push you a little bit of that question and ask you, what is the upper limit of what you can measure on the uncle?
So for intrinsic fluorescence, we say 300 mg per mil of an antibody like protein. And so that's, actually showed data here today from a 350 mg per mil antibody like protein, or antibody protein, excuse me. And so it's kind of an exciting upper limit where you get to see and analyze data using all of these tools to understand your protein.
Awesome. Thank you, Kevin. Let's see, next question. Are the B22 measurements conducted using the SLS function, or do you need a DLS for this?
So the way that we operate B22 and KD measurements is by running them both off of our dynamic light scattering laser optics. So in one experiment, you get both dynamic light scattering information and static light scattering information from the same laser and the same detector. And then if you make a choice on how to analyze that data, that's what gets you to B22 or KD information.
Excellent. So we'll take this last question. Everything else we can answer offline, just given the time. Let's see. So experimentally, which one is easier to measure, KD by DLS or B22 by SLS? And the accuracy, any method template for such analysis on the uncle? So I'm assuming someone's asking, is there an application for it, or do you have to do it manually, and how easy is it to do?
Oh yeah, so there is an application for it, and then we, as part of our, support and training, we always really, make sure that you walk away knowing how to set up your experiment. And so the idea is to run 5 different concentrations and a buffer sample, and then Uncle takes it from there and runs the analysis, gives you all the raw data you need if you want to go back and look at it more carefully, but just hands you a B22 or KD value right off the bat. And then flipping between the 2 experiments is as easy as just clicking a little radio button in the software.
Awesome. So no offline calculations, nothing else to do?
No.
Good enough. Excellent. So I think we'll wrap it up here. Lots of interesting questions, very exciting. And so we're going to follow up with your questions offline, just trying to be mindful of time. But I want to thank everyone for joining us. As a reminder, all of these videos will be available online. So if you can go to our YouTube page, Unchained Labs, you can look us up on YouTube and you will find us. You can also follow up if you have any questions at info@unchainedlabs.com. And we'll also answer your questions then. So once again, thank you. Thank you for Kevin for your wonderful and really interesting presentation. And we wish you well, and we'll see you soon. Thank you.
Thank you.